Hello guys and welcome back to Excel Share Academy. So in the previous lecture, we have discussed like how proteins and RNAs are getting transported from nucleoplasm to cytoplasm and vice versa. And we have also seen how these transport is getting regulated. Okay. Now today in this portion, in this lecture, we are going to talk about the chromatin. So yesterday in the previous lecture, I have asked a question whether the chromatins which are present, okay, does they are present in the random way or it is localized, right? So this is a question I have asked. So so both Tanvi and Akshay, they have mentioned that it is random. Did you guys get the time to go back and check whether the chromatin reorganization or organization of this is random or it is localized? Did you get chance to read about it?
I saw that it is localized.
Yeah. Yes. So it is localized. It's not random. In each cell, okay, in the nucleus, each particular chromatin has got its localized region. Now, why it is important is that, see, ऐसे imagine करो, suppose this is your nucleus, okay? So we have seen that in certain certain area of nucleus, NPCs are more, in few areas NPCs are less, right? So just imagine like how these chromatins are arranged in such a way that the access of proteins with the help of the NPC becomes easy. ठीक है, जहां पे NPC कम रहेगा वहां पे आपका chromatin structure कम रहेगा. Now why do we need this more concentration of NPC? Because the proteins which are getting translated in the cytoplasm, okay, they need to come into the nucleoplasm so that they can act as the transcription factor, RNA polymerase, DNA polymerase, etc., etc. Okay, so that's why the chromatin region. So again, we know chromatin, two types are there, right? Hetero is there and we have got euchromatin. So we know that euchromatin is the region where mainly your transcription takes place. Agree? And heterochromatin where your centromere and telomere is present. So these are always a condensed form, and here we need that the nucleosomes leave the DNA so that the transcription or any of the gene expression can take place. So these regions, the euchromatin region should be near in such a way that the proteins are accessible to this region, whereas centromere and telomere, they do not take part in the gene expression. So there the involvement of protein or the requirement of protein is less. Are you able to follow? Akshaya, understood now?
Yes, ma'am.
Yeah, okay. So let's move ahead and let's talk about these organization of chromatin in detail. So chromatin becomes highly condensed during the mitosis to form the compact metaphase chromosomes which are distinguished to daughter nuclei. Now during interphase, most of the chromatin decondense and is distributed throughout the nucleus. Now why? Because in the interphase itself, your S phase is there. That is your DNA replication takes place over here. Although interphase chromatin appears to be uniformly distributed, the chromosomes actually occupy distant regions of the nucleus and are organized such that transcriptional activity of a gene is correlated with its position. Okay. In addition, the key activities of DNA replication and transcription take place in clustered regions within the nucleus, and these clustered regions are known as factories. ठीक है, इनको हम बोलते हैं factories. We'll come to that. Now, chromosome territories. So what does it mean? It says that it just in simple meaning, it says that each chromosome has got its own territory. So it's not a random localization. Each chromosome was found to occupy a discrete region of the nucleus called the chromosome territory. Okay, so this experiment was done by the Carl Ravel in the case of the chromosomes in the salamander cells. Okay, so in the first case, what you see is the complete chromosome, and you can see that these chromosomes have got their own territory. And here in the second, they have just highlighted the telomere portion only. Okay, so what do we see here? The telomere position. So we see these telomeric positions are towards the center, right? In the nucleus towards the center. Or we have seen in the case of the nuclear membrane. Okay. Sorry. Now, Same figure with the help of the color diagram. And what the technique what they have used is FISH, that is fluorescent in situ hybridization. We'll talk about the technique in the technique section. Now this is a model of the nucleus showing over here the 5 chromosome arms in different colors. The positions of telomeres and centromeres are indicated. Now where do you see the telomeres and the centromeres positions? It is towards the inside of your or the central of the nucleus, right? Are you able to follow? Yes. Can you see? These are the telomeres. Oh, I should take another color. Here, these are the telomeres and here the centromeres. So you can see that they are present towards the inside of the nucleus. Okay. Now in this figure, they have just shown that this is one arm of the chromosome and this is the second arm of the chromosome, just to see, just to show that they're topologically separated. Okay. Now, this is another, just with the help of all those fluorescent probes, they have confirmed. So it further appears that centromere divides each chromosome into distinct domains so that the two arms of each chromosome do not interact with one another. Okay. So we know what is the function of centromere. So here you have got the centromere, right? Now, fluorescent probes to repeated sequence on chromosome 4 were hybridized to a human cell. The 2 copies of the chromosome 4 identified by yellow fluorescence occupied distinct territories in the nucleus. So what do you understand over here? इन्होंने क्या किया? पूरे chromosome 4 को fluorescently label किया. ठीक है? So wherever these fluorescent chromosome 4 was present, it shows the yellow color. So from here we see that there are 2 copies of the chromosome 4. Is this much clear? And you can see this chromosome is opened up. Okay, ऐसे खुला हुआ रखा है. Okay, any doubts over here? Are you able to imagine like how exactly these chromosomes are aligned inside the nucleus? Yes, Tanvi?
Yes, ma'am.
Okay. Now the chromatin localization and the transcriptional activity. Okay, so most of the chromatin in interphase, that is in the case of the euchromatin, it is decondensed and transcriptionally active, but in the case of heterochromatin, it is condensed and it is not getting transcribed. So ये अभी हमने just अभी discuss किया. So we know what is heterochromatin. The only the repeated sequences are present, centromeres and telomeres are there. Okay, and they are not getting transcribed. Now the transcriptional activity of chromosome domains correlates with the location in the nucleus. Now here one more thing you have to understand is that the chromosome location or the chromosome transcriptional activity is going to be different in liver cell when compared with the let us say muscle cell. Okay. Now why it is going to be different in both the cells when it is belonging to the same organism? Can anyone tell me, Shruti?
Because different proteins are required and therefore their transcription will affect the
Absolutely correct. Absolutely correct. So depending on the requirement or the function the liver is carrying out, so the protein expression is going to vary as compared to the muscle cell. And that's how the gene expression is going to vary. Hence, the transcriptional activity will be different. Very good. So transcriptionally inactive heterochromatin is frequently associated with the nuclear envelope or the periphery of the nucleolus. Okay. Whereas transcriptionally active chromatin is localized to the interior of the nucleus or adjacent to the NPC. Okay, NPC के पास है, ठीक है? Now, heterochromatin in interphase nuclei. Now, this is the again the electromicrograph over here, and what do we see that the dark color area है, those are your heterochromatin region. ठीक है? So what they are saying is that heterochromatin या फिर आपके nucleolus के पास रहेगा या फिर एकदम न्यूक्लियर एनवेलप के पास रहेगा. ठीक है? जबकि आपका यूक्रोमाटिन रीजन रहेगा एनपीसी के पास. Okay, this is again another figure. Now these microscopic observations of chromatin localizations were further done and इससे हमें पता चलेगा कि कौन से आपके active and inactive domains are there. ठीक है? For example, some human chromosomes are rich in transcribed genes whereas others are gene poor. Okay. A comparison of the territories occupied by such chromosomes indicates that gene-rich chromosomes are located in the center of the nucleus, whereas gene-poor are found at the nuclear periphery. Now, you can see it over here. The fluorescence in situ fish with the probes with the gene-rich chromosome किया है red. ये आपका रेड रीजन हो गया, ओके, एंड जो जीन पुअर हो गया वो आपके न्यूक्लियर एनवेलप के पास हो गया, ओके. नाउ अगर हमारे अभी आई डोंट थिंक वी हैव इट ओवर हियर अबाउट द एनपीसी, ओके, आई डोंट थिंक इट इज देयर, अदरवाइज what you would have seen over there that the chromatin transcriptionally active regions will be present near to the NPCs. ठीक है. Now this is for the inactive chromosomes. So again, hybridization was done for the entire mouse chromosome 2, green color, and the exon sequences of that chromosomes red. So हमने फिर से देखो, of that particular chromosomes जो inactive region है वो periphery में है और जो active region है वो आपका अंदर की तरफ है, ठीक है. Now यही क्वेश्चन अब आपसे पूछ सकते हैं. सो दिस इज नथिंग बट द सेम एक्सपेरिमेंट. इफ यू सी, फिश किया था फॉर द ट्रांसक्रिप्शनली एक्टिव एंड इनएक्टिव क्रोमोसोम्स, राइट. दिस इज जस्ट डिफरेंट फिगर, but you can see depending on the localization you understand where the active and where the inactive chromosomes are present. Okay, now the same thing I have shown how it is going to look. Same thing can be asked for the exam, like how exam in a sense मतलब interview में पूछ सकते हैं. And why I'm focusing right now again about the interview is that, as you know, the CSIR results were out and many people qualified. Right now the interviews are going on all over the India. So it's not only the CSI labs but the other JGBL's exam, for ICER, for NCBS, TIFR. They are recruiting people now for the PhD, okay, and they are taking the interviews. So NCBS, TIFR, ICER में 2 round होते हैं and the rest all only 1 round is there, but before the written exam is there. So once you clear CSIR or GGBLs, again there is an institute specific written exam and followed by the 2 interviews. So इसलिए while studying itself, get prepared for the interviews also. So एक question यहाँ पे क्या आ सकता है, like given an organism is there, okay, given all the facility, how can you tell that chromatin or the inactive part of the chromosome is present where? ठीक है, तो यहाँ पे आपको fish जो technique है उसके बारे में पूरा mention करना पड़ेगा, ठीक है. Now it will be good that if you could explain with the help of the figure. So right now what should be the answer? Suppose Akshaya, I am asking you as an interviewee, like with the help of the fish technique, how can you explain me where the inactive and the active regions of the chromosomes are present? How can you explain with the help of the diagram? Yes, Akshaya.
So where there is fluorescently labeled the green color towards the outer side.
First you have to tell me you have to tell me whether the active regions you have labeled with which color and inactive regions labeled with which color. Can you draw, like from your laptop or mobile? Can you draw?
I can try. So towards the external region, so I'm going to label with green color. So this indicates that so if I'm going to label the inactive chromosomes with suppose pink color, I am using it here, and I'm going to use for example yellow color for the transcriptionally inactive region is towards the center, and transcriptionally active region is towards the outer of the nucleus. So when I'm going to stain it and use the fluorescent in situ hybridization technique, it might indicate that when the fluorescently labeled pink color is indicating the inactive regions, and the active regions, and the yellow color ones are indicating the inactive region.
Okay, I got confused. Okay, so what I understand is that technique wise, it's clear to you, right? So in the fish, how do we do is that we use antibodies, which is like, tagged with certain fluorophore or chromophore, and we make antibodies against the proteins which are present in the chromatin proteins. Okay, so that's how you're gonna tag using the probes. Now, what I got confused is that over here, Akshaya, so you said inactive is pink and active is yellow. Kya?
I said it ulta. So the inactive is yellow and the active is pink.
Acha, no, it has to be like this. Okay, I know thoda sa confusion रहता है because what they have mentioned. Okay, when you go through again the video, शायद मैंने भी शायद एक जगह गलत बोला है now if I recollect. I'm sorry for that, for the confusion. Let's make it clear now. So inactive region is over here. ठीक है? Nuclear periphery. Active region is towards the nucleus, inside the nucleus. Can you see?
Yes.
Hybridization with the probe spanning the entire mouse chromosome to green, and only the exon sequences. Exon means what? It is the active region, correct? So that is red. Let's go and check in the another figure. Here, see. Gene-rich chromosomes are your red, which is present in the inside of the nucleus. Gene-poor chromosome is green, which is at the periphery. अभी clear है? Confusion नहीं है? Yes, Shruti, Tanvi, is it clear or still any confusion is there?
No, ma'am, it's clear.
Okay, good. So this is how, like Akshay, it was a good attempt by you. So this is how, if the questions are there. So this is, I have asked the question after explaining the entire experiment, right? But just imagine if suppose they're asking like, how do you differentiate where such proteins are present or any chromosomes are present? How are you going to do? The fish is the best technique by which you can tell about the localization of any protein inside a cell. Okay, because you just have to make antibodies against that, label it, tag them with the correct fluorophore or chromophore, and then put or just incubate the cell with that particular antibody. So kya hoga? After some time, when you see the fluorescence, you will see the localization of that particular thing. Okay. So, for example, like Shruti, you must have given the interview at Sorab's the last lab. So they must have talked about the centrosomes.
Yeah, they did.
Yeah. So what are centrosomes? So we know that inside the cell, centrosomes are responsible for formation of mitotic spindle during the mitosis. Correct?
Yes, ma'am.
They are again what? They are also proteins, right? So what they were working was that the centrosomes were labeled again, using this antibodies only. And then they saw the localization of these centrosomes here and here. So then they saw, yeah, they do get split into two. From the one is mother, the other one is daughter. Okay. Then again, they have done the experiment saying that, so we make, in the figure, if you've seen, we make centrosomes like this, 2 copies, right? So in his lab, they were working like whether this is mother, and then we get the daughter. Okay. Or is it like here mother, one daughter? Or here one daughter or one mother. So aise they have done the experiments. Now, Shruti, I'm asking you, if this is a scenario, how are you going to conduct an experiment to understand that this is both mother and then daughter, or you have got mother-daughter separation before the mitosis or the mitosis begins? So think as a researcher, use the same technique. How are you going to differentiate between the two scenarios? How are you going to confirm कि इन दोनों में से कैसा exactly होता है? Let it be whatever the reason be, but as a technique, okay, how are you going to apply your theory, your knowledge in conducting this experiment? वो कैसे होगा? That in the lab you can learn, but ये क्यों होगा? कैसे करेंगे? Like verbally, please explain. Yes, Shruti.
I'm just thinking, give me a few.
Yes. Don't take more than 1 minute.
we can use fish to fluorescently label the stones that are already present.
Okay, you're using fish to already what?
To label the histones that are present in the mother cell.
Wait, Shruti, there is no question of histones. What are centrosomes? Centrosomes में histones कैसे आएगा? Histones are present in the chromatin, in the DNA. What are centrosomes?
Centrosomes are also proteins that are used to correctly segregate the sister chromatids.
Yes, okay. जहाँ से आपका 1 second, let me draw for you. Okay, so we study like this. तो यहाँ पे आपका centrosomes आया from here, ऐसे आपके mitotic spindles आते हैं, so that they can bind to your align at the metaphase to the centromeres of the chromosomes, right? Question यहाँ पे है, so histones are only present in the DNA and nowhere else.
Yeah.
Centrosomes are your proteins which is made up of empty microtubules. It is a protein now. Okay, go ahead. अब कैसे होगा? नहीं? Not getting? Anyone else would like to try? Akshay or Tanvi?
Maybe tag the mother centrosome with one fluorescent color and the daughter centrosome with the other color. So
But
while
on
segregation.
yes, very good. But on what
Identification issue will be there because we were trying to figure out which one was mother and daughter. So
correct.
without knowing, we won't be able to.
Yes, I was coming to that point only. See, so I'll give you the answer. So your approach is correct. So Shruti, what point you have taken out now is it is a base for all your experiment, right? How will you understand the difference between the mother and the daughter cell, daughter centrosomes, right? So if you remember, whenever we talked about DNA newly synthesized from the parent and the daughter, so do you remember how the cell understands which one is the parental and which one is the daughter stand? Do you remember? Correct, methylation. In the same way, the mother centrosomes and the daughter centrosomes, they have got difference in the proteins. Okay, so when you go in detail, there is always a difference in the composition in the mother and the daughter. So the proteins are, methylated or phosphorylated or acylated. So this particular post translation modification is always there. ठीक है? तो जो mother में होगा, it won't be present in the daughter. Okay, and against that you are going to tag. You are going to make a probe. Okay, so when you do the experiment, if you see that here 2 fluorescent nuclei is coming and here nothing is coming, so what conclusion do I get? That both mother centrosomes are here, both daughter centrosomes are here. Okay, but if I get one probe over here and one over here, then I get this is mother daughter, this is mother daughter. Now you got the point?
Yes, ma'am.
Yeah, so whenever you want to differentiate between two particular proteins or two strands of DNA or anything between two, always remember that there will be some difference. Right now you don't know what is the difference. That's completely okay. Nobody can remember the differences. Okay, now once you come to the research level, you start doing, then you actually go in deep and understand like centrosomes are made up of these are the composition, this is the difference. So that is another level. Okay, no other scientist will be also knowing. But if you're in that lab, for example, the basic principle you have to remember is that whenever mother daughter parental or daughter strand, whenever we talk about, there is always a difference in their protein tags. Okay, and हम उसका use करते हैं in differentiating with the help of the fish. Clear? Clear? अभी in future अगर आपसे ये पूछा जाए कि आपके पास pancreatic cancer cell line है और आपके पास ओवेरियन कैंसर सेल लाइन है, इसके और इसके बीच में आप कैसे डिफरेंशिएट करोगे? बिकॉज अंडर द माइक्रोस्कोप बोथ लुक सेम, ठीक है, अंडर द माइक्रोस्कोप इन विट्रो बोथ द सेल्स लुक सेम. अगेन हाउ आर गोइंग टू डिफरेंशिएट? सिंपल, पैनक्रियाटिक सेल लाइंस के ऊपर जो इंटीग्रल प्रोटीन्स रहेंगे दैट इज गोइंग टू बी डिफरेंट एज कंपेयर टू द ओवेरियन. टैग दोस, उसके स्पेसिफिक प्रोटीन को आप ग्रीन टैग दे दो और ओवेरियन स्पेसिफिक प्रोटीन्स को आप रेड टैग दे दो. Look in under the fluorescent microscope. Wherever green is there, that shows it's a pancreatic cell lines. Wherever red is there, it is ovarian cancer cell lines. Of course, they might be sharing the similar proteins also, but obviously the specific proteins will be present, which gives them the characteristics of being a pancreatic cell line, giving the characteristics of being a ovarian cell line. Okay, clear?
Yeah.
Yeah, good. तो ऐसे ही हम बैक्टीरिया के साथ भी कर सकते हैं, ठीक है? ओके, लेट्स मूव अहेड. So again, this is the same, the cartoon representation of the same transcriptionally active and inactive chromosomes. So what do we see here? Again, see the inactive regions which is condensed, you can see it present near the nucleus के पास है, ठीक है, यहाँ पे present है. And then जहाँ पे आपका NPC or sorry, the nuclear envelope is there over there also. Now we also have euchromatin region जो आपके ऐसे present है in the this area. Okay, what are NAD and LAD? They are nucleolus or lamina associated proteins and domains. Okay, इतना ज़्यादा सोचने की जरूरत नहीं है. Okay, now replication and transcription factors. Let's come to that. So the functional organization of chromatin, it is further indicated by the localization of key process, which is clustered regions of the nucleus where you see all this particular DNA replication, transcription taking place. Okay, so these are known as the replication factories. Now, replication factors contain clustered sites of DNA replication within which replication of multiple DNA molecules takes place. Now, again, they have understood with the help of the fish. So here what they have done. So it's a newly replicated DNA. It was labeled by the exposure of bromodeoxyuridine, BRDU, which is incorporated into DNA in the place of thymine. Okay, so this also we have discussed when we were talking about the DNA structure. So BRDU is the analog of thymidine. Okay, yaad rakhna hai. So what you do is that you just make it fluorescent. So this substitution allows detection of newly synthesized DNA by the immunofluorescence, which is following the staining with an antibody against bromodeoxyuridine. Now, wherever you see bromodeoxyuridine, what does it show? That there is replication going on. Clear? नहीं समझ रहा क्यों ऐसे बोल रहे हैं हम लोग? It's clear, right? See, these are the regions it is incorporating the bromodeoxyuridine. Incorporating means what? Instead of thymidine, by mistake it has incorporated bromodeoxyuridine. अब वो क्यों incorporate किया? Because their replication was going on, and that's why the analog of thymidine, that is bromodeoxyuridine, got incorporated. And hence after labeling we saw that these are the regions where the red fluorescent is coming. So यानी कि ये सारी regions है जो आपके replication factories है, ठीक है? And we also know that in the case of the eukaryotic chromosomes, we don't have only one replication origin. We have got many, multiple, correct? Now, same thing they have done using the GFP labeled green DNA replication protein PCNA. Do you remember what is PCNA?
Lamp loader in eukaryotes.
Yes, absolutely correct. So wherever you have wherever the replication is going on, so PCNA will be there, correct? And it is GFP labeled. So on exposure, or sorry, on checking on the microscope, you can see that these are the points where PCNA is present. It means those are the areas where replication was going on. And this is how you are going to prove that there are replication factories. Okay, so जहाँ पे आपका replication चल रहा है. All clear? This experiment? Yeah.
Yes.
Yeah. So similarly, we have got for the transcription factories also. Okay. So similar to our replication factories, we have got the transcription factories as well. Okay. So they contain newly synthesized RNA and are highly enriched in the active RNA polymerases and the transcription factors. Okay, so it is estimated that the transcription factory contains around 8 molecules of the RNA polymerase. Several chromosomal loci can share the same transcription factory, and it is thought that active genes loop out and are pulled through a factory as transcription occurs. So in these case, they are the co-regulated genes which may be transcribed in the same factory. इसका क्या मतलब हो रहा है? Let's see the figure. That is like, you know, we'll be able to understand here more. So what do we see here? See, it is just that, see, यहाँ पे अगर आप देखोगे, 1 second. Yeah. So these are what? These are your the heterochromatin regions. Okay. These are your euchromatin regions. Now, they just loop out. जो भी region जो है आपका important है, like where the transcription should take place. So what is happening? Can you see it? They just loop out, and their RNA polymerase binds, the transcription factors binds, and they continue the transcription. ठीक है, तो यहाँ पे सिंथेसाइज होके आपको mRNA या RNA या tRNA जो भी बन रहा है वो आपका यहाँ आ जाएगा. ठीक है, are you able to imagine now? And then whatever is required according to their post transcriptional modification, mRNA में क्या होगा आपका 5 cap यहाँ लग जाएगा, 3' poly A tail यहाँ लग जाएगा, then that will be taken, and with the help of the other transporters, exporter or importers, it will be taken out in the cytoplasm. Clear? So even though right now in this figure we show only one transcription factor, so you guys remember, right? कितने सारे transcription factor लगते हैं हमें. Okay. Now able to imagine like how inside the nucleus all these things are getting arranged. Now the question comes, जैसे ही आपका mitosis चल रहा है, ठीक है? So we know that in that everything is going to get condensed. So what happens? All these regions will then again get condensed with the help of the nucleosomes. वो और जाकर condense हो जाएंगे. They will form the solenoid structure. More condense, and finally they get this structure. है ना? So that time this nuclear envelope is not present. So we have studied, right, that during the mitosis the nuclear envelope disappears. During the meta phase the chromosomes align and then get separated, and then again nuclear envelope reappear. Okay, now when you say that the nuclear envelope, okay, nuclear envelope disappear or reappear, तो यहाँ तो कोई magic नहीं हो रहा है. So when you say disappear, reappear, so exactly what is happening inside the cell? When you say disappear, matlab kya? Reappear, matlab kya? So I'm sure there is no magician present there which actually, you know, moves a band and makes it disappear or reappear. So what is exactly happening there? Why we have been taught like nuclear envelope gets disappear during the mitosis and reappear after the cell is getting divided, etc., etc.
The polymerization is taking and repolymerization is happening.
Polymerization, depolymerization of what?
Desmosomes type.
Desmosomes. Nah.
I forgot the protein that used to make the nuclear envelope. The scaffolding of that protein loses its polymerization
So
and
those,
repolymerizes after. Lamins, yeah.
Those are your lamins, correct. So these lamins, when they get phosphorylated, they actually break down. That is when it gets disappear. So disappear होना मतलब envelope जो ऐसे था अब उसके छोटे छोटे टुकड़े हो गए हैं. So they are like this, fragmented. Okay, reappear means these phosphorylation gets removed and they start getting connected to each other. So ऐसे आपका वापस आता है. So that is reappearance. ठीक है? So disappear नहीं होता है. It's just that they get fragmented and they are present. The phosphorylated lamins are present in the cytoplasm. Now once the nuclear envelope has to be present. It has to reappear. So that time the phosphorylation is being removed by the help of the phosphatase enzyme CDC25. And that time this will start forming, they will start connecting with each other, SA. So then they start forming the membrane. Okay, good. Now the nuclear body. So what are nuclear bodies? So again, they are the discrete organelles, okay, which is present in the nucleus. For example, like cytoplasmic organelles, mitochondria, nuclear bodies, they do compartmentalize the nucleus and serve to constitute proteins and RNAs for a specific nuclear process, okay. So, however, in contrast to cytoplasmic organelles, nuclear bodies are not enclosed by membranes, but are instead maintained by protein-protein and protein-RNA interactions. So, there no membrane is present. ऐसा नहीं कि nucleus के अंदर एक और जाके organelle है. It is not. It just that protein RNA interactions are there. So there is no phospholipid present. Okay. Now, so nuclear bodies are dynamic structure whose contents are able to exchange with the rest of the nucleus. So we know that they are involved in the rRNA synthesis, processing, and the ribosome production. So the nucleolus में ये काम होता है. Okay. So this is the example. So nucleolus, so around number of per nucleus 1 to 4 are there whose function is in the RNA transcription processing. Kajal bodies, clastosome, histone locus body, speckle, PML bodies, and polycomb body. So we'll talk about what is this PML and polycomb body as we progress in this lecture. Okay, ये अभी इतना important हो गया है right now because we started talking about gene silencing, interferencing, RNA, etc. तो ये सब अभी light में आ गए हैं. So it's better that we do study about this. Okay. So the nucleolus and the examples of 3 other nuclear bodies that function in regulation of gene expression and in the assembly, maturation, storage of components of the pre-mRNA splicing machineries are discussed here. So however, these roles and the many other nuclear bodies are not yet fully understood. So elucidating this aspect of structure and function of the nucleus remains an active area of research. So what I'm saying here is that many people are working on different aspects, but studying about the nuclear body is an open research topic now. So if you get a lab in future, if you have interest, then you can talk about. Nobody is studying about the nuclear bodies right now. What I feel important is that if we study nuclear bodies, we might get in way in order to target cancer cells. Like agar hum normal cell mein nuclear bodies kaise kaam kar raha hai, in the same way, if there is some difference found in the case of the cancer nuclear body, then there might be some way. I don't know. Right now, I don't have that access to the lab or to the research field. You guys, if interested, you can take up that area. Okay. Yeah, so I would like to stop here the topic and I have got very one interesting question for you guys. मैं हमेशा पूछती रहती हूँ. Few of the questions I myself don't remember to ask again in the next lecture. और तुम लोग भी याद नहीं दिलाते हो. Anyways, so the question is that you have got let us say that you have got the nucleus. ठीक है? The radius is 10 micrometer. This is all assumptions. Okay, let us imagine. उसका radius इतना है and it has got NPC around 1200 nuclear pore complex. So how or what is the distance between 2 NPC? कैसे calculate करोगे? Question समझ में आया? Should I repeat? So this is your assignment and कुछ भी हो जाए मैं ये कल पूछ के ही रहूंगी. उसके बाद ही क्लास शुरू करूंगी. So the question is, you have got the nucleus, ठीक है? I'm giving the hint, sphere. So it has got the radius at 10 micrometers.
Can we assume that the distance between 2 nuclear pores are same, like all throughout?
I'm not getting the voice clear, Shruti. Once again, can we take what?
Can we make the assumption that the nuclear pores are evenly distributed across the sphere?
Okay, evenly distributed. Yes, these are our assumptions, right? So we can take it. Okay, so the question is, nucleus है, sphere दिया है, radius दिया है, total number 1200 दिया है. I'm asking what is the distance between 2 NPC. Okay, question clear?
Yes,
Okay then. think about it, try to solve it. जिसको आ गया, please share the screenshot in the WhatsApp. हाँ, so there was this question asked by Akshay, I guess, right? मैंने वो miss किया. I'll just go through Akshay. I haven't seen the question there, and I'll let you know the answer. Maybe today in the WhatsApp chat, or then tomorrow I'll discuss. Okay, sorry, I was busy. Okay, so I hope this question is clear to everyone. आज के लिए अच्छा question दिया है. So see the conditions now. Even though you study bio, but without physics, chemistry, maths, it's difficult to crack the written exam and crack the interview. So अगर किसी ने ये सोचा था कि that हमने बायो लेके हम इन सब चीजों से बच गए हैं, so sorry to say no. Okay.